Right, thank you for coming. Guess I have the task of keeping you awake after lunch. I'm going to try it. So just quickly, I'm going to start by talking a little bit about plant domestication and agricultural history and how where my scientific interests lie in this topic. Then I'm going to talk about the work I've been doing on the domestication of rye and how rye manages to adapt to such different environments as Scandinavia and Morocco. And finally, I'm going to finish by talking a little bit about the genomic analysis of museum collections of rye grains and how we are using that to assess changes in biodiversity in this crop. Well, this being a research institute in agronomy, I don't think I need to explain how the emergence of agriculture was one of the major events in human evolution, probably paired only by bipedism and learning how to use fire. I mean, it brought about an astonishingly change in the way humans live, and especially a change in human population numbers, a trend that is definitely still ongoing with its dramatic consequences in terms of sustainability and ecosystems worldwide. But the process of domestication also brought profound changes in plant genomes and their phenotypes as individuals with desirable traits amongst a population of diverse wild plants, particularly individuals with traits like the seeds being retained in the ear or huge fruit size were selected by the first farmers, and that brought about profound changes in the plant genomes as well. And it is this process of plant domestication and how the interaction between humans and plants changed plant genomes that I'm very interested in, and in assessing how much biodiversity has changed throughout these human-plant interactions. I don't know if anyone has ever been to St. Petersburg. I was there 2 years ago, and I had the chance to visit not only the Hermitage, but also the Russian Museum. And this was a painting I saw there, and I thought this was rather interesting. It's from 1948, typical social realism. It's slightly better than Malevich squares and Golden Virgin Marys, which is legion in this museum, but still strongly recommend visiting this museum. It's wonderful. But what it really fascinates me is that this painting depicts one of the later stages of crop evolution. And what I want you to notice is the height of the wheat plant. Okay, this is typical Russian scientists finding ways to improve wheat and feed the people, etc. But look at the height of the plants reaching shoulder length. And this is a theme we see in a lot of paintings throughout history. Famous Bruegel, The Harvesters, Egyptian tombs, medieval Book of Hours. Notice how the plants are always so tall. And this is rather strange because if you go out to a field today, this is what you'll see. The plant will probably reach shoulder length. And why is this so different? Why is wheat today so different from what we see in the art? And of course, this has to do with the fact that from the 1950s onwards, plant varieties that we grow today, commercial varieties, have been introgressed with a gene that reduces the plant height. So it makes it less susceptible to tiling, makes it easier to harvest, and more importantly, it allows the plant to channel nutrients into grain filling, providing not only more abundant yields, but also more nutritious plants. And these varieties are so widespread, these commercial varieties are so widespread, that, that what we see today is just that short wheat, totally different from the things we see in the arts. And until the mid-19th century, cereals cultivated throughout, well, at least throughout Europe, they were cultivated pretty much in the same way. The farmers had a bunch of seeds that they would, that were locally adapted, so that they've been growing it for ages, and these were called land races. And this is a very important concept I'm going to be coming back about to. So varieties of a crop that are perfectly well adapted to particular locations, and they might not fare well in different locations. The farmers would pick a bit of grains from a field, right? The ones that fare better. Oh, this one is slightly better because it's taller or has a better grain or didn't succumb to diseases. So I'm going to use this and sow it in the next season. And they've been doing this for years. But from the late 19th century onwards, what happened is that plant breeding companies came along and they started producing high-yield varieties, which included varieties with desired traits like resisting to diseases or producing high yields. And this is not a bad thing, of course it's not. I mean, it really allowed feeding many more people. But as farmers abandoned these land races and adopted these varieties, these commercial breeds, what we saw was a reduction in biodiversity. And this has been quantified in several crops, actually, how this change from land races to industrial varieties really reduced the gene pool available within crops. And this genetic uniformity really poses a threat to the sustainability of agriculture. But if you think about it, all agriculture is about reducing genetic diversity. I mean, if you look at the stages of plant domestication, first there were wild plants, like in this case wild wheat, and they were extremely diverse, okay? As people domesticated plants, they selected only a few individuals, the one with the desired traits, and they domesticated those. They kept cultivating those. So immediately you have a genetic bottleneck, okay? A lot of diversity, a lot of alleles of different genes got reduced into just a few varieties of those genes. As agriculture spread, plants went adapting to different locations, and this allowed a slight increase in diversity worldwide of crops. Okay, so things were getting slightly better. Then 19th century comes along, we start producing very well adapted varieties, very high-yield, that will die out completely in a bad year, but never mind, because worldwide it works, but you have a worldwide reduction of diversity. So all in all, do you remember this scene in the movie Gladiator where Russell Crowe's character goes very onerically passing his palms through the wheat? This is just a big fat anachronism. I mean, this in Roman times, the guy would have to pull up his gladium and slash his way through the wheat. Speaking of Romans, the Romans were great agronomists. I mean, they left us amazing texts on agronomy like Columella and Varro and Pliny, telling us a lot about the variety of crops that the Romans had. And one of them was rye. So the Romans are the first that registered that they were actually cultivating rye, and we see that in some archaeological excavations. Nevertheless, if we believe Pliny, the Romans didn't really like rye. They say it was very disagreeable, not so good for the stomach. They were not the biggest fans. But in the 5th century, a few centuries later, when Pliny wrote, the Roman Empire collapsed. I mean, it must have been dramatic. An empire that was everything that people knew in those days completely collapsed. It was followed in the 6th century by a dramatic coolness of the weather in Europe. It was very, very cold. There were diseases going around. The Romans had developed amazing agricultural systems with irrigation, and throughout about 6 centuries. By this time, the soils were becoming salinized, so all that irrigation was starting to fail, and probably there were major crop failures all throughout the empire. And it is in this period the rye really saw an increase in cultivation, becoming the major crop during the Middle Ages in a lot of European regions. I mean, in Scandinavia, it was the major crop up until the beginning of the 20th century, really, when wheat, again, that wheat production finally made it more, the yields be better for wheat than they were for rye. And the reason for why rye probably took off during this time is that rye is a rather interesting crop. It tolerates salinity much better and can produce good yields in poor soils and conditions that other cereals probably couldn't do. But unlike other cereals like wheat and barley, whose process of domestication has been very well studied from the archaeological point of view, from the genetics point of view, we don't really know very much about rye. We don't even know for sure what the wild ancestor was, or even if rye was domesticated only once, or in different time periods, in different places. There's still a lot we don't know. What we do know is that it probably spread as a weed. It kind of resembles wheat and barley, okay? So probably the first farmers were growing wheat and barley, and they saw that strange ear there, but they thought, you can actually eat this, it's not bad for you, so why should we bother rooting it out? So they just tolerated it. And it's very interesting because in archaeological sites, prehistoric archaeological sites in Europe, when agriculture is coming into Europe, what you see is that the seeds that show up in the dig are mostly wheat and barley, okay? Rye shows up as only as a very small proportion, 1–2% about-ish. And they only become, we know that rye was being cultivated for its own sake much later on, because we start having sites where everything is rye in the archaeological seeds recovered. So we know that rye was really becoming important. So in a way, rye is really interesting because it's a case of plant mimicry, which is not very common, and definitely not in crops. I don't know many cases of crops mimicking stuff and being selected because of that. The archaeological record suggests that rye first appears about 10,000 years ago in the Fertile Crescent area there in pink, which is basically where most crops in the Old World appeared. And it later on found its way into Europe again, probably as a weed and not necessarily as a crop in its own right. Rye thrives in moist conditions and it thrives very well in cold conditions. And it might not be a coincidence that the stages of rye becoming more important are associated with cold spells. Again, look at here. This is about the time when the Roman Empire fell. Cold spell in Europe, and then rye really takes off. Late Middle Age, the weather gets better, wheat replaces rye. Might be a coincidence, but it might just be a strategy of farming communities facing changes in the climate to just, wheat and barley are failing, let's just grow rye. It's not the best thing in the world that keeps us alive. So actually makes good bread. I never quite understood why Pliny didn't like it. Also, in contrast with most plants we have studied domestication, like wheat and barley, rye is an outbreeder. It makes it very interesting because when we look at wheat and barley, what we know is that it was it's very easy to domesticate it. You select a plant that's different, he has the characteristics you want, and you just grow it. There's not going to be any pollen mix, anything. The plant is going to stay faithful to its phenotype. It's very interesting to look at rye because it's not like that. I mean, it will change because of pollen intercrosses. It's been more or less studied in maize, which is the same breeding system, but maize was domesticated in America, so it's a whole different story. So rye is rather interesting from this point of view. Also, like most of crops, its taxonomy is just hell. I mean, it's really, you can see there's a lot of stuff. There's feral rye, there's wild rye, there's the cultivated rye, and they all look slightly different. And this is actually important. I'm going to be talking a lot about this. You have, within the Secale genus, you have the cultivated, the Secale cereale, okay, on top. This is the rye we grow. Then we have the wild rye here, that are suspected that were the wild ancestor. I mean, it was probably after these wild plants that rye acquired those characteristics that make it domesticated, okay? The two typical suspects are Secale strictum and Secale vavilovi. All of them occur in the Fertile Crescent area. Then there are some Secales that we know because they're really different and they're self-pollinating, so they probably had nothing to do. They did not contribute to the gene pool of the cultivated rye. And these are the Africanum and the Silvestri. And then you have the feral rye, Afghanicum, Sagittale, Dicoricum, Ancestrale, plenty of those varieties that they're probably runaways from cultivation. Fields that got abandoned, seeds that got taken away, and then they grow in the hedges and become something completely different and strange. So this is more or less what we wanted to look at. We wanted to look at how rye was domesticated. So how many times was it domesticated? Can we know that? Can we know the wild ancestor? Can we know the genetic relationships between the wild and feral and cultivated rice? I mean, was there a lot of genes going around by introgression, going from wild to cultivated and to feral, etc.? More importantly, is there population structure in cultivated rye? I mean, is rye just a uniform crop that looks pretty much the same everywhere? Or do we definitely have different gene pools where cultivated rye is very different? Also interesting, we wanted to know which genes were actually involved in giving cultivated rye its characteristics, and which genes underlie the fact that rye can grow as well in Scandinavia as in Morocco. I mean, which genes are involved in these? For this purpose, we had 52 populations of cultivated Secale cereal, mostly land races. So again, those varieties that were well adapted to different locations from most of Europe, including Portuguese land races. We had 5 to 6 individuals per population because we also wanted to study how diverse the different land races are within population diversity, as opposed to between population diversity, okay? And we compared that with 25 populations of wild and feral rice, again 5 or 6 individuals per population. The genetic markers we used to quantify and compare genetic diversity in these populations were SNPs, single nucleotide polymorphisms, which are just point mutations along the genome that differentiate between individuals. Like in this case, one individual will have a T allele in that point there, and the other one would have an A allele. Just to give you a highlight of how this worked, we had a panel of 769 SNPs. At the time I started this, there were only 4,000 SNPs published in rye. I don't think there's many more now. There's a lack of genomic tools for this crop, unfortunately. And this is a very small number. If you think of it, this kind of studies with humans, they go up to sometimes 10 million SNPs. Which makes this pathetically small, but it's what we had. We got seeds sent to us from germplasm banks of land races and wild varieties, so I did not go to the fields to actually pick them up. The seed banks sent them to me. We germinated them, did DNA extractions, sent it to using the Illumina Golden Gate to detect the SNPs, and then what we have is something like this. So each line is an individual plant, okay? And these are individuals of the same population, okay? And each column is a SNP. And what you can see just from this image is, can anyone see? Can people see this? Okay. Is that there is, even within the same population, you see different individuals have different genotypes. And what we do is we quantify the frequency. For example, for this first SNP, the frequency of A's and C's in the different populations, and that allows us to compare ancestry, to compare populations, and to quantify how much diversity there is. These are the results we had. So this is just a principal component analysis based on the allele frequencies of all these things, and each point is an accession, okay? The triangles are the wild rye, the squares are the feral rye, and the circles are the cultivated rye. And the first thing we see is that we have these wild ryes here, which are Strictum and Africanum, and they're very far apart from the cultivated points, okay? On the other hand, we have these wild guys here, Secale vavilovi, and they are not, I mean, there's one here as well. They really cluster well with the cultivated varieties. Then we have the feral types, which some of them are quite separated from the cultivated types, but some of them are really close, like in this case, that case there, which seems to suggest two things. One, that Strictum and Africanum had not really contributed any genes to the cultivated stock, so they are not the ancestral of rye, whereas Vavilovi probably is the ancestor of the domesticated rye. And that the feral types, like in this case and that case there, they really are runaways. I mean, they probably descend very clearly from cultivated rye, but they just became different because the selective pressure of farmers to grow it was abandoned. So they just went feral as it is. Okay, is anyone familiar with structure? Structure is a software that's used in population genetics. This is actually quite complicated to explain. I'm going to try to do it. But imagine we want to compare how rye in Portugal is, if it descends or how genetically close it is from rye in Spain, rye in France, Italy, etc. Okay? Back in the day, you'd probably do a phylogenetic tree, something like Portugal, is this writing? Portugal, Spain, France, Italy, no, something like this would come along. Okay. But there's actually no, I mean, we're doing a lot of assumptions that might not be correct here. Why should the rye in Portugal be the same as the rye in Spain? I mean, think of it as people. If we take randomly 100 people in Portugal and 100 people in Spain and 100 people in France, why shouldn't the Portuguese and the Spanish be the same from population, from a genetic point of view? So there's these assumptions that structure gets away from. It's actually a modeling software, and what it does, it takes the allele frequency of each individual plant and populations, and then it computes, you tell it, give me 2 populations, and it divides all the individuals into 2 populations, and then 3 populations, and 4 and 5. And then it tells you, well, the most likely is 5 populations, and it clusters the individuals into these populations that we can call ancestral populations, and they might or might not fit into the categories that we were thinking. So for example, here the software tells me that with all my accession of my collection of individuals, he finds 9 populations, okay, 5, he finds 9 distinct gene clusters as it is, okay? And let me start trying to explain this. These things, so each vertical line, okay, you see there's a lot of vertical lines. Each vertical line is an individual. And it just shows the proportion of alleles it gets from each of these 9 populations. Is everyone following? So for example, if you look to this here, there's actually 5 individual or 6 individual vertical lines. And most of them get their alleles from a yellow population, okay? Whereas these guys here get it from a blue ancestral population, so different from the other ones. Is this more or less? Okay. So I also put them together by the land races, which are this, well, it doesn't matter, but they're clustered by land races, and then by species. Okay. So we're looking at 3 levels here: the level of the individual, the level of the land race, and the level of the taxon. Is this making sense? What we observe is that in the cultivated land races, we find 5 gene clusters, okay, 5 populations. A light blue one, a red, a dark blue, a pinkish here just by 2 accessions, and the yellow ones. Okay, so there's 5 distinct gene clusters in the cultivated rye. We also see that these 3 here are commercial breeds that I included in my set, and we see that they do get their alleles from different populations. So it's probably just telling us where the plant breeders went to pick up their parental individuals to do the crosses. But thing to keep in mind, 5 cultivated gene pools in rye. So rye is not a uniform crop. There's this substantial diversity there. When we look at the wilds, we see that they mostly cluster in this dark green thing here, okay? Except Vavilovi, that has a lot of the dark blue, etc. Now this, I'm going to try to explain this. In the strictum, we see there's a light blue and a purple individual, okay? And this might connect to the ferals or cultivated. When I first did this, I thought, interesting. It means that strictum was also, the wild strictum was also contributing to the cultivated stock, just like Vavilovi is. Then I actually started to cultivate this, and when they got to ear stage, I realized that these two things, they're not strictum at all. So it's a misclassification of the seed bank. They were cultivated rye. So note, be advised, do not trust completely in germplasm banks when you do these kind of things. They were IPKs, by the way. I didn't tell them this because they would be upset. But and then in the feral rye, you have some ferals that are clearly related to the cultivated. Like, look at this orange here. This variety is from Afghanistan, okay? Cultivated rye from Afghanistan, and it gets alleles from the same pool as Afghanicum, which is clearly from Afghanistan. And then there are some feral rye that have no connection whatsoever to the cultivated, like this purple here. Okay, so they seem to be something on its own. Hope I managed to more or less explain this sort of Pollock kind of thing. I then took only my cultivated land races, okay, and I did the same software. By the way, this uses Bayesian statistics, and it takes about a month to run in the computer, so on a modern computer. So it's actually mathematically quite demanding to model these things. But when I did this with a cultivated rye, I get 5 clusters, which means that, yeah, it fits what I had before. Again, 5 gene pools in the rye. And when I run it in the wilds and the ferals, again, I see that now excluding those two bizarre mislabelings, I find that Africanum and Strictum have no connection whatsoever to the cultivated rye, but that Vavilovi and then all the feral ones do seem to stem from a common pool. Okay, now this is the geographic distribution of the cultivated rye land races. So forget all the wilds and the ferals, this is just the land races. And because we know the geographic provenance of these land races, we can actually point them in a map. And what these pie charts are showing is that each pie chart is an accession, and it's just a proportion of alleles that they get from those ancestral populations. Is this making sense? And what we see is that we have a distinct group of accessions, the light blue ones. They're very typical in the Middle East, Turkey, and a little bit in the Balkans. Okay, so they occur there. The yellow ones, Scandinavia. They're very typical in Scandinavia, a bit in Central Europe. Dark blue ones, Western Europe, all the way up to Scotland, France, Portugal, Morocco, Spanish rye. They fit into this population. Then there's that pink, funky one there in Norway, which is really interesting because those pink ones, they're not even, I mean, they're land races, but the way they cultivate it is by slash and burn. They just bring down the forest, burn it, spread some rye, and let it grow. It's not even, they don't even go there to, I mean, it's called slash and burn. It's just a kind of semi-wild cultivation type. But what this is showing is that there are distinct populations in Europe occurring. Okay. This is actually quite relevant, not just from a historic point of view, which means that they might have different people bringing rye to different regions, but it's also interesting for plant breeding, which means if you want to maximize your likelihood of having different gene combinations, don't go mixing a Portuguese with a Moroccan landrace because it's the same population. Try mixing a Moroccan with a Scandinavian, and that will increase your novel combination of genes. Interesting, the rice from Portugal, they're very diverse. This guy here is from Guarda, and it's a funky one. It probably means that Portuguese rye is very mixed. Table with numbers, ignore the numbers, don't be scared. I just want you to look at this column here, okay, which is the heterozygosity, which is an unbiased measure of genetic diversity. Okay? It's not affected by how many individuals I sampled in each of these populations. And this is actually a bit tricky because of ascertainment bias effects with SNPs, but I'm not going to go into that. But it needs a bit of caution. But in any case, what we observe is that there's a lot of diversity in the cultivated rye, more than in the wild, okay, and that is ascertainment bias. All the literature says that wild needs to be more diverse, and it probably is. The problem is that the SNPs were identified by genotyping two elite breeds, okay, and finding polymorphisms between them. This does not mean that these polymorphisms are the same in wild rye. So when you try to use a panel for breeds to screen diversity in the wilds, you will get a difference, which does not affect population structure, but it affects quantification of genetic diversity. But in any case, it's still interesting to see that Ancestrale seems to have more diversity than the cultivated cereal as well, which is interesting. And when we compare different geographical regions, we see that there isn't that much difference between the rye. So rye, although there are different populations of rye, as I showed in the map, the genetic diversity in these populations is not that different. And yet more numbers, never mind it. This is just a table showing FST and genetic distance. And this just shows how different the different taxons will be from each other, okay? And what I want you to realize is that for both FST and genetic distance, the lowest differentiation and the lowest genetic difference is between Secale vavilovi and Secale cereale. So if you remember, vavilovi was the suspected ancestral. Okay, so all this is pointing towards, yeah, vavilovi is, in fact, the ancestor of the cultivated stock. And strictum always fares very, very distant from all the other things. So strictum is ruled out as a likely ancestor. The next thing I did was trying to find out which genes were affected during the process of domestication. So lots of things change when a plant is domesticated. The plant size, the fact that the seeds get retained in the year, the uniformity in time of sprouting, etc., etc. And there's a big scientific enterprise to try to find out which genes are actually controlling these phenotypes. And I try to do this by looking at FST, which is again a measure of differentiation between populations, and heterozygosity. In this case, each dot is an allele, okay? It's just a version of the SNPs. We're not looking at individuals here. So just alleles with a particular value of diversity and a particular value of difference between the cultivated and the wild rice, okay? And what I want to find out is outliers. When we find outliers, like this guys in red here, it probably means that these alleles were under selection. Think of it this way. If you have an allele that's being very strongly selected against or for, then it's going to have a different frequency in a population than a different allele in a population that has not been under that strong pressure. Is this making sense? Actually, I had a picture to explain this better, but oh well. Anyway, when I looked at the flanking regions of those SNPs, and I blasted it to try to find out a function of what these SNPs were doing, surprise, surprise, most of them had no information because the genome of rye is not annotated, and it's incredibly hard to find homology to other genomes that are properly annotated. Nevertheless, there are some interesting results, like this SNP here that was one of those outliers is actually a transcription initiation factor. And this has been, in other species, it's been associated, a lot of the genes underlying domestication have been transcription factors. So genes that actually affect a lot of gene expression in different things, which allows domestication to happen very quickly. So instead of changing genes that affect individual characteristics, sometimes you have a mutation in a transcription factor that's going to have a cascade of changes in the plant. So this is actually interesting. Then there's a lot of genes that I would love to try to do a functional genomics study on them to see if they actually do anything like RNA interference or silencing these genes, but I don't think I'm going to be doing that anytime soon, but still there's some clues about genes being affected. The other thing I was really interested about is, look, how does rye grow in Scandinavia and in Morocco? This was really intriguing me. Which genes are affecting this adaptation? And to try to find this out, I did a different thing. I looked at, again, the frequency of alleles in the set of Scandinavian land races and compared it with the frequency in the Iberian and Moroccan land races, so north against south. And the idea, again, is that if you have an allele that gives the plant a good adaptation to cold, then in Scandinavia it's going to become very frequent, whereas in the south it's going to be less frequent. Okay? So what I have here is that comparison of, again, each dot is an allele, not an individual. And what I wanted to find out was, again, the outliers. So most SNPs will have, that's a very beautiful trend line, because most SNPs will have the same frequency. I mean, they won't be much different. They're just rare or frequent in the whole overall population. But then you have those strange things. One of them is really interesting. It's a potassium ion channel in Setaria. So again, I blasted the flanking sequences to try to find a function. It might hint at adaptation to salinity being affected somehow in this adaptation process. But again, it's just a clue. Another one associated with glycolysis, which is rather interesting. So it might be that underlying adaptation to this different environment is actually profound metabolic changes in the plant itself. But this again remains a bit speculative. Okay, now if you remember, I mentioned how biodiversity changed along the transition from wild to cultivated, and then from land races to the commercial breeds that we grow today. And one of the things that I wanted to look at was how rye's agrobiodiversity changed in the last 150 years. So in that period where the land races were abandoned and the commercial breeds came along. And I had a fantastic opportunity to do this. And rye, before I go into that, rye is rather interesting because it has not been subdued to profound pressure from breeding companies. I mean, they're more interested in maize or rice or wheat. Rye has been a bit of overlooked by the breeding companies. And so in a lot of places, land races are still cultivated. And for example, if you see here, this is a rye land race, and this is my 2-meter-tall Swedish boss. And as you can see, the freaking plant grows really tall. And these are images from a 16th century Swedish book on folklore and culture, etc. And you can see that already then the rye was quite tall. Accidentally, the book also tells you that if you're harvesting, you should hang your baby cribs from trees, or the snakes will come and snatch the babies, which is rather interesting. But we had a great opportunity to look at these changes in biodiversity because we had access to the Nordiska Museum in Stockholm, which is a museum of all things related to Nordic culture. It's a fascinating museum. If you ever go to Stockholm, I mean, just the building is mind-blowing. It's in the central island in Stockholm, strongly recommend it. And they have, well, first of all, they had a lot of these flasks, hundreds and hundreds of them, which were the leftovers of a world exhibit in 1860, I think, where they sent varieties from all over the place to there, and then they all ended up in the Royal Swedish School of Agriculture, and they were used for teaching purposes. The really interesting thing is that we have the record of the year where they were harvested, and they come from different plants. So we know that they just went to the bag and got them. So this is heaven for population genetics. I mean, you have something, it's a genetic snapshot in time. I mean, you can go there and if you get DNA out of it, and look at how things were in 1860s. They also have a lot of these beautiful straw goats, which are typical Swedish artifacts. They use it in their Christmas artifacts, and they do it all over the place in Sweden. And some of these were from 1840s up to 1920s, 1930s, and they were all there in the museum. So we can go and actually go. They were made with rye straw, typically, and the beards of the goat is rye ear. So it's just really nice to go there, pick it up, get DNA out of it and do some studies. The first thing we actually went to do is to see if DNA survived in this. So we used ancient DNA methods, a lot of precautions with contaminations, and it's a bit complicated, but we did find that there was a lot of DNA. DNA was amazingly well preserved in this. This is just a PCR of a chloroplast marker, microsatellite, and we see that this is a modern control, and it amplifies almost as well as the modern controls. Although some samples, like this one here, you see there's a faint band here for this marker, which is 105 base pairs. But if you go up to 130, then it fails. Mind you that the DNA is there, but it's very fragmented. I mean, you can amplify it up to 200, 300 base pairs. Anything more than that, and it's not possible to look at it. But if you use SNPs, it's really cool, because the way SNPs work, it's usually 50 base pair fragments anyway. So it makes it very interesting to use in these materials. And what we did was we looked at 191 of these rye grains from different parts of Sweden and Norway and Finland and Germany as well. And we selected 55 of those SNPs that we had typed before, so the results I showed you before. We chose the ones that were most polymorphic, so to maximize the information we could get for these historical seeds. And we went to screen the SNPs in there, in these historical collections, and then compare it with the results we already had for the land races from the same regions. I'm not going to go very deep into this. It's just on the left, you have historical varieties, okay, the results for the historic accessions, and on the right, of modern equivalents that we analyzed coming from seed banks. And the first thing, I mean, it's a bit eyeballing, really. It's not very statistically accurate, but we see that here, the elite breeds and the land races were, oh, I forgot to say, we also had access to some of the first rye commercial breeds that were being produced at the Svalov breeding company in Sweden in the late 19th century. So it could actually trace nicely the transition from land races to breeds. And we see here that at the late 19th century, they were not that different. Things are more or less mixed. The red are elite breeds, the blue historics. Whereas when we look at the modern counterparts, we see that things are not really as well mixed. Whereas when we look at it by countries, again, we see that things are not really changing that much. I mean, we don't clearly see breeds here and land races there. So, which I think it's good news. It means that the biodiversity in rye is not really being dramatically affected by plant breeding. Although this is a bit worrying how land races that came from germplasm banks are being so diverged apart, whereas here they actually seem pretty close. This might have to do with the way that germplasm banks work by regenerating things every 25 years or so, which creates bottleneck after bottleneck after bottleneck. So might be a bit worrying. And again, this is interesting because you don't see, even in the modern parts, you don't see elite breeds, okay, being very different from the land races. Okay, they're more or less closer together. Now compare these with the same methods I used in a paper for wheat. This is the one that got published last week. Yay! And what we see, look at the green dots are wheat commercial breeds, and the blue here are land races. And look at the difference. I mean, you clearly see the breeds separating from the land races. So the effect of plant breeding seems to be more intense in wheat than it is in rye. And just to finish, quantifying the genetic diversity for those 55 SNPs in all the materials we studied, again, being a bit cautious with ascertainment bias effect that keep telling that wild is less diverse than the cultivated, but it still works if you're comparing only rye. And what we see is that historical land races and historic elite breeds and elite breed, there has been a decrease in genetic diversity, but it's very small. I mean, Historic landrace to landrace today, and I mean it's very, very small, which seems to suggest, one, that plant breeding in rye is not really affecting its overall biodiversity, which is good news, and that the germplasm banks are doing a good job. I mean, if they're, with all the issues associated with the work, they seem to actually be doing a good job, at least when it comes to rye. And just to conclude, there is population structure in rye. Okay, rye is not uniform. This is good news. There's very different gene clusters in the landrace rye that can be used for plant breeding and to study historical movements of people and trade. Secale vavilovi is probably the only ancestor of cultivated rye. It's probably the only wild species that actually contributed to the gene pool of cultivated rye, and that although rye is an open pollinator, there does not seem to be a lot of gene flow going around from the wild rye to the cultivated, which is interesting. Using these methods, we can probably identify genetic differences or the genes that explain the domestication syndrome and adaptation, but it definitely needs a lot more work, and it needs probably 1 million more SNPs to actually do this type of... I keep dreaming of doing a GWAS study with rye, but I don't think it's going to happen anytime soon. But there definitely there's clues that domestication and adaptation, we can trace the genes that are doing it, which is good news. And finally, that agrobiodiversity in rye has not been significantly reduced by the breeding programs. So it is good news. I think we don't have to worry too much about rye biodiversity. It's still a very diverse crop, especially if you look at land races, which are a treasure vault for breeding companies that should probably be better used. And just to finish, just some words of acknowledgement, and thank you.